Humans have cultivated cannabis sativa for thousands of years. With recent legalization and increased interest in research, medical, and industrial applications, cultivation of cannabis now presents a high-value agricultural commodity. Australian medicinal cannabis patients are prescribed through the Therapeutic Goods Administration Special Access Scheme Category B. Prescriptions are granted on an individual basis to commercial formulations or cultivar products of specific cannabinoid content. Medicinal cannabis products must conform to stringent sourcing, quality, and composition requirements outlined within Therapeutic Goods Order 93 to ensure consistent active ingredients. This includes the need to appropriately source and identify cannabis varietals. Therefore, securing legitimate, stable phenotypes for consistent cannabinoid production is of great importance. Classification and identification of cannabis sativa cultivars is challenging. Patients and prescribers rely on informal industry distinction of cannabis sativa, hybrid, and cannabis indica, despite the fact that immense crossbreeding and informal cultivation practices have resulted in a vast landscape of hybridized cannabis cultivars, possessing little taxonomic difference, yet large variation in cannabinoid and terpenoid content. Chemotypic grouping of cannabis cultivars through analysis of genetic markers and cannabinoid-terpenoid profiling provides a more accurate distinction needed for patient reliability, consistency, and industry reform. This is supported through gas chromatography analysis of TGO 93 compliant products to ensure consistent active ingredients between batches. But does cannabinoid content vary between phenotypic expressions of cultivars, and can source phenotypes exhibit the consistency required for TGO 93 compliant production? I conducted analysis on 3 distinct phenotypes of OG Kush cultivars and 1 distantly related Afghan Kush landrace to determine if a significant difference in the mean cannabinoid concentration of phenotypes could be found. OG Kush phenotypes and Afghan Kush are prescribed to Australian patients. OG Kush is a cannabis sativa dominant hybrid, ANTG Mariposa. Afghan Kush is a cannabis indica cultivar, Medcan CC01. For relevance to local context, phenotype populations were selected from corresponding cultivars. A large dataset detailing chemical analysis of commercially available cannabis cultivars was obtained from a University of Boulder, Colorado study. From this dataset, a selection of 3 populations of OG Kush phenotypes, including OG Kush, Tahoe OG, and Fire OG, was selected alongside a distantly related landrace strain, Afghan Kush. ANOVA was selected as appropriate analysis for comparing more than 2 groups of means, with analysis conducted on mean CBGA and THCA levels to determine if a statistically significant link between cannabinoid content and cultivar could be found. ANOVA of CBGA content presented a significant difference in the average CBGA concentration in flowers in OGK phenotypes and Afghan landrace cultivars. The greatest mean CBGA concentration was found in Afghan Kush varietals at 1.72%, over 3 times higher than OG Kush phenotypes, which present no significant difference in CBGA concentration. ANOVA of THCA contents presents a significant difference in the average THCA concentration found in Afghan landrace cultivar AK. The highest THCA concentration was found in OG Kush phenotypes OGK and TOG, with FOG phenotype presenting no significant difference in THCA concentration compared to AK. Cannabis affects humans through an endogenous neural network called the endocannabinoid system, where plant-produced cannabinoids such as tetrahydrocannabinolic acid, THCA, cannabidiolic acid, CBDA, and cannabidiolic acid, CBGA, selectively bind to CB1 and CB2 receptors, promoting homeostasis and providing a range of therapeutic properties. Cannabinoid and terpenoid biosynthesis is modulated through gene expression of cultivars. CBGA is a precursor to other cannabinoids and plays a primary role in THCA biosynthesis, and thus determining final cannabinoid concentration. Analysis determined that OG Kush phenotypes exhibit consistency in cannabinoid concentration, inferring relative genetic stability, while Afghan Kush varietals exhibit a statistical difference in both CBGA and THCA concentrations against OG phenotypes, least pronounced in those of immediate Afghan genetics, both FOG and TOG, inferring a possible distinction in biosynthetic pathways between distantly related cultivars. Based on analysis, OG Kush phenotypes present consistent cannabinoid concentration and would be suitable for TGO 93 compliant production. They also significantly differ from related cultivars, further highlighting the challenges of broader cultivar classification. Despite the challenges of classification and sourcing stable phenotypes of cultivars, GC analysis to determine consistency of active compounds among phenotypes provides a reasonable starting point in identifying and grouping cannabis varietals to ensure TGA compliance. However, industry terminology requires reform to ensure consistent patient outcomes.